Today, I'm going to talk about hand washing. This is experimental lab, the effect of hand washing on the bacterial contamination. Microorganisms can be found or are found everywhere environment, on the surfaces, in the air, and on the human body. The discovery of the importance of hand and skin surface disinfection in disease prevention is credit to the Semmelweiss at Vienna General Hospital in 1846. Semmelweiss established a policy of hand washing that resulted in a drop in the death rate due to puerperal sepsis from 12% to 1.2 in 1 year. That means it's around 11% drop in the death of that case. Although the skin is never completely sanitized, never. The transit and normal flora can be significantly reduced by prolonged scrubbing with soap. Correct hand washing is the single most effective technique that prevents the transmission of microorganisms either directly or indirectly to others. It is essential to show that the hand washing method does in fact just remove microorganisms from the skin and will not kill them. To test the hand washing technique, fluorescent dye is applied to the hands prior or before hand washing. Following washing, the hands are examined under UV light for any traces of dye. Compare your hand washing with the diagram that shows commonly missed areas. We'll find out in that missed areas in the following diagrams. Here, they applied before hand washing with fluorescent dye. Then wash the hands. Then after that, subjected the wash the hands with fluorescent dye and see the traces of dyes on the fingers here and here. There's not be there. If you compare this one and this one, this is with the red color is here, this area and this area, and between fingers is mostly frequently missed. Most of the time is missed. With little dark brown is frequently missed, like this way and this way in these areas. This is the front hand, this back hands, you see. And less frequently missed these areas, here, 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 and here. You see, these mostly frequent missed here. Material, what we need for this lab? We need hand soap, 9 nutrient agar plates, hand sanitizer, disposable hand drying paper. Procedure, we have around 13 steps. Number one, step number one, label 9 the nutrient agar plates into 1 through 9. Plate 1, or step 2, plate 1 is a negative control. Don't touch it and attempt not to pass your hands inside this plate as you complete the exercise. Step number 3, touch agar in plate 2 with and wash it 4 finger tips. This will be the positive control. Rinse your hands with just tap water for 20 seconds. Dry hands and touch finger, 4 finger tips to plate number 3. Wash your hands with tap water and liquid soap for 20 seconds. Dry your hands and touch 4 finger tips in the plate number 4. Step number 6, wash your hands with tap water, soap for 20 seconds, and sanitize them. Dry again your hands and touch 4 fingertips in the plate number 5. Step number 7, wash your hands with tap water, soap for 40 seconds and sanitize them. Dry your hands and touch 4 fingertips in the plate number 6. Step number 8, a sample of liquid soap sprayed on the nutrient agar in the plate number 7. As a swab from a bar of salt soap also sprayed into nutrient agar in the plate number 8. To find the effect of sanitizing solution on the bacteria, sneeze on the nutrient agar plate 9 and then a drop of sanitizing solution in the middle of it. Incubate all the plates at 37 degrees centigrade for 24 hours. Examine the plates for relative abundance and diversity of growth. Record your results. Results. Plate shows a negative control results. This, we don't touch it, we don't put our fingers, we do nothing. You see, it has no microbial at all. This negative, what they call negative control. Positive control, we put, or they put their fingers or tips of fingertips on the plate agar and incubated at 37 degrees centigrade for 24 hours. This is mean unwashed hands. This is mean dirty fingers touch agar. Results after incubation period, you will see bacteria growth after 24 hours due to the contamination of unwashed hands. The dirty unwashed fingers contain so many bacteria that the areas they touched on the agar plate were a mass of colonies. You see, mass of colonies. You see how many? These unwashed, dirty fingers, authentic fingertips, unwashed fingers after incubation after 24 hours. Bacterial growth after 24 hours due to contamination of rinsed hands, washed hands with water only for 20 seconds. See, there is not that much reduced bacteria. Water will remove nothing, just a little, you know, but there is still a lot of it, lot of growth. This number five, figure number five, bacterial growth after 24 hours due to contamination of washed hands with water only. If you compare both of them, this is unwashed fingers, these washed fingers with water. You see, mostly not that much reduction. It's mostly the same here or here. Wash your hands with tap water and liquid soap for 20 seconds. Washing the hands with soap and water for 20 seconds reduces the number of bacteria, but it was not long enough removing them all. You see, still with soap for 20 seconds, still bacteria in your fingers or finger tips, still there, a lot of them. Not that, not they reduced, not that, but reduced how many? It's about 67%, 30%, but still about 70% or 60 or 70% is present. The contamination is there, still there in your fingers. If after the hands were washed with soap and water for 40 seconds, the number of bacteria decreases even more. Recommendation methods say wash your hands for 21 seconds, but 21 seconds will not remove all of them. Even 40 seconds will not remove all of them. It's still there after 40 seconds. This agar plate shows that washing hands with soap and water for at least 40 seconds will reduce the number of bacteria on your hands, will reduce. Washing them for a full minute works even better. Even though removing all bacteria is impossible with soap and water alone, the important thing is to reduce the number of transient bacteria on your hands. Always be sure to wash your hands thoroughly for at least 40 seconds before you handle food. Shows for 40 seconds, this figure numbers are fingertips after an additional 20 seconds wash with soap and water. No bacteria grow on the agar plate after the finger were dipped in the sanitizing solution. The bacteria may or may not have been killed, but the sanitizer stopped their growing. Of course, some people may not be able to use sanitizing solution because it may irritate their skins. You see, this is washing hands with water, soap for 20 seconds and sanitizing them. There is no growth at all, you know. Washing with water, soap, and sanitizing hands or fingers, there is no growth at all. These finger Tips after using a sanitized solution, there's no growth at all. You see, no growth at all. A sample of liquid soap sprayed onto nutrient agar and a swab from a bar of solid soap also sprayed onto nutrient agar. We'll find the results. Soap are not designed to kill microbes, just to remove them, not to kill them. They help to wash them off your skin better than water alone. Why do you think that the solid soap kept by the skin by sink and handled regularly had more bacteria living on it than the liquid soap kept in dispenser, so not handled, which had none? You may be interested to know that while soaps do not kill microbes, they can be quite a good medium for them to grow on. This is plate agar sprayed on liquid soap. There is no growth. Incubate for 24 hours at 37 degrees centigrade. There is no growth at all. This is a swab from a bar solid soap and and so for so also spread on the agar media or plate agar media. There's growth after 24 hours at 37 degrees centigrade. There is nothing zero with liquid, and it with solid soap or bar look solid soap is very high contaminated. This is the effect of liquid on solid soap and bacteria, liquid and solid soap on bacteria growth. The incubated agar plate shows the effect sanitizing agent on bacteria growth. An agar plate was sanitized on, and then a drop of sanitizing solution was put in the center of the plate. Bacteria grow in the area not touched by the sanitizer. See, this is sneeze it in the plate. We dropped one drop of sanitizing solution and incubate for 24 hours. Then we find there is the area of sanitizing area. The border strikes no growth, but others no sanitizing agent. They grow, bacteria grow very well, and this is highly contaminated. How many bacteria in new sneezing found? Reference. Thank you for watching my video about hand washing, and goodbye.